Large-scale poultry production is a challenging business that can be jeopardized by devastating infectious diseases, such as Newcastle disease and avian influenza. Avian influenza virus is a particularly potent threat as it continuously mutates in the field, resulting in divergent strains with altered surface proteins, a process known as antigenic drift. In countries with endemic disease, vaccination is the preferred method for prevention. Conventional inactivated vaccines and live recombinant vaccines are both widely used. However, both have their limitations. For instance, while inactivated vaccines provide adequate protection against existing strains, they may not protect against newly mutated viruses. Exposure to these new strains can result in disease and mortality. Live recombinant vaccines were developed as an updated alternative to inactivated vaccines. However, they may incur an extended period of vulnerability to infection between vaccination and the onset of immunity. What then is the optimal avian influenza vaccine? It would protect against a broad spectrum of virus types, inducing a rapid and strong immune response, with minimal side effects, and could be produced on a large scale. Additionally, it would facilitate distinction between naturally infected and vaccinated birds, a strategy known as DIVA. Boehringer Ingelheim, a world leader in animal health, has accomplished this using a highly antigenic surface protein of the avian influenza virus that has been explicitly developed for superior protection. For this engineering process, Boehringer Ingelheim deploys a powerful and versatile technology: Baculovirus Expression System Technology, or BEST. BEST utilizes the baculovirus, a vector virus that only infects insect cells. Using the BEST methodology, a foreign gene encoding for the desired antigenic surface protein is inserted into the genome of a baculovirus. This baculovirus is cultured in specific insect cells which translate the gene into recombinant protein. After incubation, the cells are harvested and the perfectly structured protein is extracted. The advantages of BEST are many. Since the baculovirus only infects insect cells, it is very safe for both the environment and for the operator. Post-translational modification of the protein can be incorporated, and vaccines with high antigenic yields can be produced rapidly in large batches. Using the BEST technology, Boehringer Ingelheim has developed a convenient combined vaccine containing hemagglutinin subtype 5 of the influenza virus, or HA5. Hemagglutinin in general plays a key role in binding the virus to its host cell. Antibodies resulting from vaccination efficiently inhibit attachment of the virus. This blocks the infection process. During the development of Volvac BEST, the gene sequence for HA5 was specifically engineered before it was included in the baculovirus genome. These modifications ensure that Volvac BEST AI plus ND brings vaccination against avian influenza to a superior level. Studies show that the antibodies produced by the modified HA5 in Volvac BEST protect against a broad spectrum of influenza viruses, comprising strains of different generations from multiple geographical areas. Volvac BEST AI plus ND is combined with the inactivated ND virus, providing comprehensive protection against another prevalent threat, Newcastle disease. The inclusion of the highly effective ND component in Volvac BEST AI plus ND is a powerful defense against two major risks to the global poultry industry. Volvac BEST AI plus ND, because prevention works.